Welcome to the deep dive. Today we're stepping into the world of disease detectives.
That's right.
We're taking a close look at Chapter 36 from Prescott's Microbiology, all about epidemiology and public health.
Hmm.
Our mission really is to get a handle on how we track, measure, and ultimately control infectious disease outbreaks. Understand the toolkit.
Exactly. And that toolkit belongs to epidemiology. Comes from the Greek, meaning study upon the people. It's the science, the evidence-based science, looking at how health and disease happen, what causes them, where they spread, and how we control them in, well, specific groups of people.
So epidemiologists are the detectives, basically.
You could say that, yeah. They're hunting for clues, for those factors that let a disease take hold and spread.
And who are the big players coordinating this? I know internationally it's the WHO, the World Health Organization.
Correct. Based in Geneva. And here in the U.S., it's primarily the CDC, the Centers for Disease Control and Prevention down in Atlanta.
And this idea of public health isn't exactly new, is it? I remember reading about the Marine Hospital Act way back in 1798.
That's a great point. It shows this isn't a modern invention. Public health really grew out of necessity, tackling huge epidemics like cholera, smallpox. These were major threats.
But it's just as critical now with things like Ebola, SARS, AIDS, influenza.
Absolutely vital. The principles are timeless, even if the pathogens change.
Okay, so if we're going to talk like these disease detectives, we need the vocabulary. How do they classify disease frequency? It's not all just outbreak, right?
No. There's a scale. At the lowest level, you have sporadic disease. It just pops up occasionally, irregularly. Think of maybe bacterial meningitis, some cases here and there.
Yeah, okay.
Then there's endemic disease. That's the constant low-level presence we sort of expect. The common cold is a classic example, always around.
Right, background noise almost.
Kind of. Now, if that baseline starts to creep up, maybe the common cold gets worse in winter, but isn't a full-blown epidemic yet. We might call it hyperendemic. It's increasing, but gradually.
Got it. And then things get more serious. Outbreak, epidemic, pandemic. What's the key difference there? Scale.
Pretty much, yeah. An outbreak is sudden, unexpected, but often limited geographically or to a specific group. Like, remember the measles outbreak tied to Disneyland in 2014? That was an outbreak.
Okay.
An epidemic is when the number of cases shoots up suddenly, way above what you normally expect for that area of population, lots of people getting sick at once. And we always look for the index case, the first identified person in that epidemic chain.
The patient zero, so to speak.
Essentially, yes.
Right.
And then the biggest scale is pandemic. That's an epidemic that's gone global, spreading across large populations, usually defined as crossing borders into at least 2 countries or continents.
Like the 1918 Spanish flu or the 2009 H1N1 swine flu.
Exactly those, textbook pandemics.
And you mentioned the history. The origins of tracking this stuff go way back, don't they? John Snow.
John Snow. You really can't talk epidemiology without him.
Yep.
London, mid-1800s. Cholera is rampant.
Everyone thought it was miasma, right? Bad air.
That was the prevailing theory, yes. Miasma. But Snow, he was skeptical. He didn't just accept it. He started mapping.
Use mapping.
Mapping where the deaths were occurring. He noticed a huge cluster of cases around one particular water pump on Broad Street. He analyzed the data, the locations.
And realized it wasn't the air.
It was the water. Contaminated water from that pump, his big intervention. He persuaded the local authorities to remove the handle from the pump.
Just took the handle off?
Yep. And the cholera cases in that area dropped dramatically. He used data, observation, not the prevailing theory, to find the source and stop the spread. That's foundational epidemiology right there.
That's incredible. And that same detective work continues today, just with better tools, I guess, like the CDC tracing those salmonella outbreaks.
Exactly. In 2011 and 2014, they used DNA fingerprinting, a much more modern tool than Snow had, obviously.
Right.
They traced these widespread salmonella infections back to, believe it or not, specific strains commonly used in microbiology teaching labs.
Wow. How did they get out?
It seems basic hygiene lapses were likely involved. People, maybe students or staff, weren't washing hands properly after handling the microbes. It really drove home how critical basic hygiene is, even in controlled environments.
So this constant monitoring, this detective work, that's public health surveillance.
That's the term, yes. It's proactive, ongoing. We monitor everything: genetic factors in the population, environmental conditions, people's behaviors, the infectious agents themselves, even things like antimicrobial resistance patterns.
All to spot risks early.
To spot risks, understand trends, and ultimately protect the population's health.
You can really see the impact. If you compare causes of death now versus, say, 1900 in the U.S.
Oh, it's dramatic. In 1900, infectious diseases like pneumonia, tuberculosis, influenza were top killers. Big ones.
Yeah.
By 2016, the list is dominated by chronic metabolic diseases, heart disease, cancer. That shift is largely thanks to public health successes. Sanitation, vaccines, antibiotics, surveillance played a huge role in guiding those efforts.
And the way surveillance is done has changed too, hasn't it? Less about forced quarantines now.
Definitely. Pre-1940s, it could be quite intrusive, inspections, quarantines, things like that. The modern approach is much more about monitoring disease progress, gathering data, and sharing information. It's about partnership with healthcare providers and the public. We get data from required clinical reports, lab results, disease registries, even surveys out in the field.
Makes sense. But to make sense of that data, you need the right measurements. How do epidemiologists actually quantify disease frequency?
It relies on good counts, first off. You need to know the total population size, how many people were potentially exposed, and how many actually got sick.
Right.
Then we use key statistical measures. Two really fundamental ones are incidence and prevalence.
Okay, what's the difference?
Incidence measures the new cases appearing during a specific time period compared to the healthy population that was at risk. It tells you the rate at which the disease is occurring. Think of it like the speed of spread. It reflects risk over time.
So a high incidence means it's spreading fast right now.
Exactly. Prevalence, on the other hand, is a snapshot. It's the total number of people infected at a single point in time, regardless of when they got sick. So prevalence depends on how fast new cases are occurring, insidations, and how long people stay sick.
So prevalence is more about the overall burden of the disease at this moment.
That's a good way to put it. It reflects the total number of active cases. Then we have rates that tell us about outcomes. The morbidity rate is basically the sickness rate, new cases during a period divided by the total population, often expressed per 100,000 people.
Like 700 cases per 100,000 is 0.7% morbidity.
Precisely. And mortality rate is the death rate, the number of deaths from a disease divided by the total number of people who had that disease. Tells you how deadly it is.
And technology's helping map this stuff too, right? You hear about remote sensing and GIS?
Oh yeah, remote sensing, RS, using satellites, sensors combined with geographic information systems, GIS, is huge. It lets us layer disease data onto maps with environmental info.
Like what?
Things like elevation, rainfall, vegetation type, temperature. So say you're tracking malaria, you can map mosquito breeding grounds based on water bodies and climate data. Or for Lyme disease, you map tick habitats using forest cover and humidity predictions from satellite data. It helps predict where risk is highest.
So you can target interventions better.
Exactly. Makes public health much more precise.
Okay. So once an epidemic is underway,
Yeah.
figuring out how it's spreading is crucial. You mentioned patterns, right?
Yeah.
Common source versus propagated.
Yes. The shape of the epidemic curve, the graph of cases over time, tells you a lot. A common source epidemic has a characteristic look. A sharp, rapid rise in cases, a peak, and then a fairly quick decline, maybe over a week or two.
Why so fast?
Because everyone gets exposed to the single contaminated source around the same time. Think food poisoning from one bad batch of potato salad at a picnic. Boom, lots of cases quickly.
Gotcha. And the other type? Propagated.
Propagated epidemics look different. The curve rises much more slowly, it's more prolonged, and then it gradually declines. This indicates person-to-person spread.
Ah, so one person infects a few, they infect a few more.
Exactly. It takes time for the disease to move through the susceptible population, one incubation period after another. Strep throat or flu spreading through school, that's often a propagated pattern.
Understanding that pattern must really dictate the response needed.
Absolutely. For a common source, you find and eliminate the source. For propagated, you need isolation, maybe quarantine, vaccination, if available things, to break the chain of person-to-person transmission.
Which leads us nicely to herd immunity. Such an important concept.
Hugely important. Herd immunity isn't about individual immunity, it's a population-level defense. When a high enough percentage of people in a community are immune to a pathogen, usually through vaccination or having recovered from infection,
The pathogen can't spread easily.
right, it hits dead ends. There aren't enough susceptible people close together for it to maintain transmission. This indirectly protects the susceptible individuals who can't be immunized in babies too young, people with compromised immune systems.
But there's a tipping point, isn't there? The threshold density.
Yes, that's the critical factor. The threshold density is the minimum number or density of susceptible individuals needed for a pathogen to keep spreading. If the number of susceptible people drops below that threshold, the disease transmission slows or stops.
But if it rises above that threshold?
Then herd immunity weakens or breaks down. An endemic disease might suddenly become epidemic because there are enough susceptible hosts to fuel transmission again. This is why maintaining high vaccination rates is so crucial.
And sometimes the danger isn't even someone who looks sick. The classic example is Typhoid Mary, right?
Mary Mallon, yes, a truly famous and tragic case. She was a cook in New York in the early 1900s. She carried Salmonella enterica serovar typhi, the bacterium that causes typhoid fever, but she herself had no symptoms. She was an asymptomatic carrier.
And she spread it unknowingly.
Completely unknowingly. As she moved from job to job, cooking for different families and institutions, she left a trail of typhoid cases behind her. She was eventually linked to at least 53 cases and 3 deaths. It highlighted the major public health challenge of carriers who can spread disease without being sick themselves.
A sobering reminder. And we also have to remember the pathogens themselves aren't standing still. Influenza is notorious for changing. Antigenic drift and shift.
Yeah, influenza is the master of disguise. Antigenic drift refers to the small, gradual changes, minor mutations that accumulate in the virus's surface proteins over time.
Is that why we need a new flu shot every year?
That's exactly why. Drift means the virus slowly changes, so last year's immunity might not fully protect against this year's slightly different strains. We update the vaccine to try and match the currently circulating variants.
Okay, so drift is gradual. What's antigenic shift? That sounds more dramatic.
It is dramatic. Antigenic shift is an abrupt, major change in the influenza virus. It usually happens when 2 different flu strains, say a human strain and an avian bird strain, infect the same host cell, maybe in a pig.
And they swap genes.
They can reassort their genetic segments, creating a completely novel virus subtype with surface proteins that human immune systems haven't seen before. Think of the jump to H3N2 years ago, or the potential for other major shifts.
And because it's novel.
The population has little to no pre-existing immunity. That's what can trigger a pandemic. Antigenic shift is the big one we worry about for widespread, severe outbreaks.
So despite past successes, we're facing new challenges. You hear about emerging and re-emerging diseases all the time now: Zika, Ebola, MERS, not to mention antibiotic-resistant bacteria like MRSA. Why this apparent increase?
It's a complex, a mix of factors, really. Human factors are huge. We live in denser populations, more urbanization. Global travel is unprecedented. You can be exposed to something on one continent and be on another before symptoms even start. That collapses the geographic barriers that once slowed spread.
And biomedical stuff like transplants.
That plays a role too, creating susceptible populations. Then there are environmental factors. Climate change alters habitats for vectors like mosquitoes and ticks. Deforestation and moving into wild areas increases contact between humans and animal reservoirs of potential pathogens.
Like Ebola, jumping from bats.
That's a prime example. And our interconnected world means problems spread. Misuse of antibiotics in one country can drive resistance that becomes a global threat very quickly. Everything's linked.
And one specific environment where these threats converge is the hospital itself. Healthcare-associated infections, or HAIs.
Yes, HAIs, sometimes called nosocomial infections. These are infections patients get while receiving treatment for other conditions in a healthcare setting. Hospitals, clinics, nursing homes.
How common are they?
Alarmingly common. The CDC figures suggest maybe 5 to 10% of hospital patients acquire an HAI. That's potentially 1.7 million infections a year in the U.S. alone.
Wow, and the cost?
Huge. Billions of dollars annually. 28 to 33 billion is one estimate. It's a massive burden on the system and, of course, on the patients.
Where do these infections come from? The staff? Equipment?
Both. Sources can be animate staff, other patients, visitors who might carry pathogens, or inanimate contaminated medical equipment, surfaces, even things like hospital water systems or food.
What are the most common types of HAIs?
Looking at the proportions, urinary tract infections, often linked to catheters, are usually the most frequent, maybe around 30%. Then surgical site infections, bloodstream infections, and pneumonia are also major categories.
And the microbes causing them. Are they superbugs from the start?
Not always. Often they're bacteria that are part of our normal microbiota, usually harmless on the skin or in the gut. But in a compromised hospital patient, or if they get into the wrong place, like the bloodstream or urinary tract, they can cause serious infection.
And the real danger is resistance, right?
That's the critical issue. The hospital environment, with its heavy antibiotic use, acts like a pressure cooker, selecting for resistant strains. So we see MRSA methicillin-resistant Staph aureus, and VRE, vancomycin-resistant enterococci.
Nasty stuff.
Very. And increasingly, we're battling highly resistant gram-negative bacteria, too, like Pseudomonas aeruginosa or Enterobacteriaceae, that produce enzymes called extended-spectrum beta-lactamases, making them resistant to many common antibiotics. They're a major challenge.
Okay, so faced with evolving pathogens and resistant bugs, what are the main strategies for control? You said break the chain at the weakest link.
That's the goal. Public health interventions generally fall into three broad categories based on where they interrupt the cycle.
Okay, type 1.
Type 1, reduce or eliminate the source or reservoir. This is about targeting where the pathogen lives or originates, isolating infectious individuals like they tried with Mary Mallon, culling infected animal reservoirs, treating sewage, and ensuring safe drinking water.
Makes sense. Type 2.
Type 2, break the connection between the source and susceptible people. This involves general sanitation measures, chlorinating water, pasteurizing milk, proper food handling inspections, vector control like spraying for mosquitoes that carry malaria or West Nile virus, basically interrupting the transmission route.
And type 3 must be about us, the hosts.
Exactly. Type 3, reduce susceptibility and raise herd immunity. This is primarily achieved through immunization, vaccination. Also includes things like prophylactic treatment, giving preventative medicine to those at high risk.
And vaccines are really the star player here, aren't they?
Arguably the single most cost-effective public health tool we have. Ever since Edward Jenner used cowpox to protect against smallpox back in 1798, vaccines have been revolutionary. They prime our immune system, creating antibodies and memory cells.
Now, there are different kinds of vaccines. The older ones use whole cells, right? Killed or weakened.
Correct. Whole cell inactivated, or killed, vaccines use pathogens killed by heat or chemicals. They're generally safe because they can't cause disease, but they often don't provoke as strong or long-lasting immunity, so you might need multiple booster shots.
And live ones.
Whole cell attenuated or live vaccines use a weakened living form of the pathogen. They usually give very strong, often lifelong immunity with just 1 or 2 doses because they mimic a natural infection. They stimulate both antibody, humoral, and D-cell cellular immunity effectively.
But there's a small risk.
A very small theoretical risk, mainly for severely immunocompromised people, that the weakened pathogen could cause illness. Or, extremely rarely, it could potentially mutate back towards a more virulent form like we've occasionally seen with vaccine-derived polio.
So to avoid those risks, newer vaccines often don't use the whole bug, like a cellular or subunit vaccines.
Exactly. These use just specific parts of the pathogen-purified proteins, or inactivated toxins called toxoids, or antigens made using recombinant DNA technology. They focus the immune response on key targets and avoid the risks associated with whole cells, much safer profile.
And the really cutting-edge stuff is DNA vaccines. How do those work?
It's a fascinating approach. Instead of injecting a protein or a whole microbe, you inject a small piece of the pathogen's DNA, usually engineered into a plasmid.
DNA directly into the muscle.
Yep. Your own muscle cells take up this DNA and temporarily start producing the pathogen's protein encoded by that DNA. Your immune system sees this foreign protein and mounts a response, both antibodies and killer T cells.
Wow. Any big advantages?
Potentially several. They seem to generate a really broad immune response, and importantly, DNA is very stable. These vaccines often don't need the strict the cold chain that many traditional vaccines require, which is a huge plus for global distribution.
Now, besides natural outbreaks, there's the intentional threat, bioterrorism.
Unfortunately, yes. That's the deliberate use of biological agents, bacteria, viruses, fungi, or their toxins to cause disease, death, and fear in a population.
We've seen attempts, right? Like salmonella put on salad bars or the anthrax letters.
Those are key examples. Bacillus anthracis spores sent through the mail in 2001 really highlighted the threat.
Why would someone choose biological agents?
They have certain terrifying advantages for a terrorist. They're invisible, odorless, tasteless. Detection is difficult initially. Symptoms are often delayed, meaning the agent can spread widely before anyone realizes an attack has occurred. This delay also fuels panic and uncertainty.
And the potential impact is huge.
Experts estimate that even a small amount, say a few kilograms, of effectively dispersed anthrax spores could potentially cause casualties on a scale comparable to a small nuclear device, mainly due to the difficulty in treating inhalation anthrax once symptoms begin.
So how is the public health system prepared for this? I've heard about select agents.
Right. There's a list of select agents and toxins that have the potential to pose a severe threat to public health and safety. They're tightly regulated. Within that, Tier 1 agents are considered the highest risk. They're highly infectious, easily spread or weaponized, and cause severe disease. Anthrax is a Tier 1 agent.
And how would an attack likely be detected?
The first sign would probably be doctors in emergency rooms seeing a sudden cluster of unusual illnesses, maybe something not normally found in that region, or presenting with atypical symptoms. This is where the Laboratory Response Network, LRN, comes in.
What's that?
It's a network of labs across the country, local, state, federal, trained and equipped to quickly detect and confirm biological threats. Sentinel labs, the local ones, would do initial tests. If they suspect something like anthrax, they send it up the chain for confirmation. Rapid detection and communication are key.
Okay, wow. We've covered a lot of ground here. Let's try and boil this down. What are the big takeaways from this deep dive into epidemiology and public health?
I think there are four main points that really stand out. First, epidemiology is the bedrock. It gives us the tools like incidence, prevalence, morbidity to actually measure and monitor health in populations, understand risk, and guide action. It's fundamental.
Right. Metrics matter.
Second, disease doesn't spread randomly. It follows patterns. Recognizing whether an epidemic is common source or propagated tells us how it's moving, and therefore what kind of interventions are most likely to work. The pattern dictates the response.
Okay. Patterns guide action. Third takeaway.
Herd immunity. It's not just about individual protection, it's a community shield. But it's fragile and depends on maintaining that threshold density through high vaccination coverage to protect everyone, especially the most vulnerable. It needs active upkeep.
Herd immunity as a community effort. And finally, number 4.
The threats are dynamic and ongoing. Our interconnected world, pathogen evolution like antigenic shift, growing antibiotic resistance. These mean that emerging diseases and HAIs aren't going away. We need constant vigilance, robust surveillance, and continued innovation in things like vaccine technology, like those DNA vaccines, just to keep pace.
Constant vigilance, constant innovation. Makes sense.
So maybe a final thought to leave everyone with, something to chew on from the chapter.
Okay.
We talked about antigenic shift creating potentially novel viruses, and we know antibiotic resistance is eroding our treatment options. How does this challenge that core public health concept of maintaining threshold density for herd immunity? If our tools, both preventative vaccines and treatments, become less effective against rapidly evolving bugs, what bigger, maybe systemic changes do we need beyond just individual choices like getting vaccinated to truly stay ahead of microbial evolution on a global scale?
That's a heavy question. A lot to think about there about where we go next. Definitely food for thought. Thank you for being a part of our little last-minute lecture family.